Okay, so last time we left off on this right here. All right, so what is a chiral center again?
All 4 points are different.
Yeah, we have 4 points that are different around a carbon, right? And so a chiral center is like saying handedness, right? You have a right and left hand, they're non-superimposable, they're mirror images of each other, right? And so this right here is not a chiral center at all, is it? Okay? This is what's referred to as a Fisher projection. It's the way you will typically see sugars listed, okay? And it's because it makes it very easy to identify which sugar that is. So you always put the most oxidized carbon up at top. You'll notice the most oxidized carbon typically isn't fully written out. You just put like the O and the H right on it without any indication of what direction it is, because it's not a chiral center. It just doesn't matter. Okay? Now, the next one's down. You're going to list it straight down where the bonds between the carbons are aligned vertically. Can everybody see it okay? Okay. These bonds right here are going away from you. So if you were to look at this chiral center right here, these bonds are going away from you. Okay? So this carbon and this carbon are actually back behind the plane of the projector. Is that making sense? Can everybody sort of see that? So the whole thing actually goes like this. Okay? And it's kind of like a ringish shape, or a C. Right? In fact, it actually would be wrapping back into a ring. Okay? 99% of it would exist like that in blood. This right here, then, is going to be a chiral center. It's the carbon. These bonds are actually coming what direction? Out towards you, right? They're jutting out towards you. And so what we would have this and that, right? And that gives you a tetrahedral shape, doesn't it? And so these are each tetrahedral shapes. And that makes that ring like that, with these all jutting out. That making sense? Can everybody kind of see that in their head? Okay, fantastic. Makes it very, very easy then to identify chiral centers, because the hydrogen is on this side, the OH is on this side. That gives you one chirality. If you were to flip-flop those, that would give you the other chirality, wouldn't it? Yeah. So if the hydrogen on this side, the OH on that side, it would give you the opposite chirality. Okay? All right, so this one down here, that's also not a chiral center, is it? No, why is that not a chiral center?
There's 2 hydrogens.
Yeah, 2 hydrogens, right? Not 4 different things, like we had heard before, which is absolutely correct. And so again, these are all going to be tetrahedral shapes, but in this case, the exact chirality, it doesn't have chirality, right? It's achiral. It doesn't matter which side the hydrogen is on. That freely rotates, it doesn't matter. Okay. And so this is actually glucose. When you see a sugar that has the OH on the right side here, OH on the left, and then OH on the right here, so right, left, right, right, that is glucose. There's also another form of glucose where you have switched all of them completely. So you flip all of these. Okay? And those are the D and L forms of glucose. This one is D. Okay? Do you know how I know that? The last one. That's how it's labeled in terms of sugars. So D is always the right side, L is always the left. And that's it. Okay, so let's look at some different forms here. All right, so right here you see that we have here the same OH. Then we have this third one here, OH on this side. And then we have this OH here. And can everybody see that we have the different chiralities for each of these different groups here? Okay, can you see the parts of your sugar? Okay, fantastic. All right. So again, the number of stereoisomers possible is going to increase with the number of chiral centers that are present in the molecule, because we can flip-flop each of these different groups, right? If we don't flip up all of them, it will no longer be glucose. So if we were to flip all of these, that would be the opposite enantiomer of glucose. Okay? But what would happen if we just flip one? That's no longer glucose, is it? Yeah, it would no longer be a mirror image, would it? It would be like me having my other hand and this finger's turned around. That would no longer be a mirror image of my hand, right? You'd be very concerned for me, wouldn't you? Okay. All right, so molecules with one chiral center, there's only going to be two possible stereoisomers that exist. That general formula for the stereoisomers is always going to be 2, because there's 2 possible for each position. And then N, where N is the number of chiral centers. And that would be the number of arrangements that you can have. Anybody that does anything with computers or has done some stats work will recognize this formula because that's the binary formula. Okay? Because there's only 2 options, which makes it binary. Because the glucose has 4 chiral centers, you can actually have 16 stereoisomers. Only one of these stereoisomers is our preferred energy source. The rest of these stereoisomers are actually different types of sugars and would actually get converted in your body if you were to use it to power your brain. Okay? So it's quite interesting. So again, the Fisher projections, horizontal lines are the ones that are going to represent your wedges, those horizontal lines. The vertical lines are going to be the ones that represent your dashes. Those are going away from you, right? The chiral center is typically not shown as a C because you will know that by the fact that the lines cross, right? This just gives a very easy, quick way to tell the difference between these. The designation of D or L is going to be based always on the Fisher projection, positioning of the glyceraldehyde, and this can be used to reference the molecule for the designation. So you're always going to base it on the Fisher projection. So again, this would be the wedge dash version. This right here is the Fisher projection. Is that confusing anybody? Everybody's okay? Okay. Fantastic. So this right here is actually the ball stick model oriented so you can kind of see the 3-dimensional structure of this. And everybody can see that this one here is in the plane. These 2 things are popping out towards you. This right here is going away. Okay. All right, and so we see here, this is the direct mirror image of the Fisher projections. You'll notice there's only 1 chiral center in this case, and so there's only 2 enantiomers, right? There's only 2 different things you can have. Okay? 2 conformations here. Now, again, these would be mirror images of each other, right? And so they are the enantiomers of each other. They have the L-glyceraldehyde. Notice that, again, the glyceraldehyde is OH, the last OH. It's on the left side. Over here, we have the D-glyceraldehyde. The last OH here is on the right side. Okay? Does this position matter here in terms of like OH is over here? No, this has nothing to do with anything, right? You could have written the OH over here on this. It does not matter. It's just not chiral, right? This one, does it have any, do we care we wrote the O over here, the H over here? Is that something that matters? No. It doesn't matter at all, right? Because again, that's not chiral, okay? Its right and left positions just don't matter. However, what does matter is right here, we have the most oxidized carbon, which is the aldehyde in this case, the carbonyl, is on the top, okay? This one, which is the last carbon with the hydroxide on it, is on the bottom. And then all of the OH, the water substitutions, are on the right and left. Can everybody see it okay? Okay. That is important. You always need to have your carbonyl pointing up top. Okay? All right. Does anybody know why that would be? What would happen if I flip this 180 degrees? Right becomes left, left becomes right, doesn't it? Yeah. So if I rotate this, all of my groups would flip. Okay, so D sugars, these are going to have the OH on the chiral carbon, farthest from the carbonyl, on the right side of your molecule. The enantiomer with the L sugar is going to have that OH, that last one, last chiral carbon, on the left side. The L sugars are the easier one to remember. L4, left. D4, right. By the way, in case you're wondering, this is actually, if I remember correctly, this is German, actually. Yeah. A lot of this chirality, and in terms of structures of molecules, was actually done back when Germany was the big chemistry power of the world and did all the research. Fun fact, that's how Blitzkrieg worked. They had a form of meth speed that the soldiers would take. So Germany was very, very good at organic chemistry back then. In a terrible, terrible way. And yes, they were all addicted to essentially meth. Yeah. So, I know. All right, so this is going to be most carbohydrates are found in nature. The ones we use for energy are typically the D sugars. And so you'll see, for example, D-glucose is the one that we use as our primary energy source. It's the only sugar we use for our brain, and it powers our brain. So when we draw these enantiomers in Fischer projections, we're going to write them as if there's a mirror placed between the two molecules. We'll attach the atoms or groups on the right side of the enantiomer that appear on the left side of the other. So what you're going to do is, here's our D-glucose that we've been talking about this entire time. Everybody see that okay? Fantastic. So what we're going to do is we're just going to take this, rewrite that part, and then we're going to take the OH, and it's going to appear anywhere it's on the right, it's going to end up on the left over here. Okay? Anywhere it's on the left, it would end up on the right side. Because we have to go ahead and flip all of the H's and OH's. Okay. Everybody good with that so far? Right. Anybody have any questions? Okay. It's fairly easy to do, right? All right. So let's talk about drawing an enantiomer in a Fischer projection. So again, you got to locate the chiral centers. Again, which ones are chiral centers? This one. No, this one. Yes, this one. Yeah, you guys got it. Okay. You're going to switch the horizontal groups on the chiral centers. So everywhere you have a chiral center, you switch them. To be honest, it doesn't actually matter if you switch to these two, because it just wouldn't matter, right? But by convention, you don't do that because it's just silly. Be extra work. Okay, let's talk about stereoisomers that are not enantiomers. So there's our stereoisomers that are going to not be enantiomers. Those are referred to as diisomers. Okay? So for example, we have these ones from our previous D-galactose and L-galactose here. These right here are enantiomers. They're mirror images of each other that are non-superimposable. Okay? And so those are enantiomers. These right here, see how they have different names and not just L and D? That's because they are not enantiomers. Okay? They're diisomers. Right? Think of it as like di. You have two options for each chiral center, right? And then stereoisomers, right? So diisomers are going to have not all of the groups flipped from each other. And so they will no longer be mirror images of each other, but they will no longer be the same stereoisomer. They will no longer have the same configuration, right? They will have different positioning. Okay? Okay, so three-dimensionally they are different. Can everybody see where these are different? Okay, so we go here, and this is the same here, right? This one, on the other hand, is also the same. This one right here is the one that's different, isn't it? Okay. And then we have the same here, right? And so we've only flipped one of those groups, and that makes it diisomer. Okay? Specifically, this would actually be glucose, this is lactose, this is talose. Those are the sugars. Right here we are D, D, and D, because they all have OH on the right side. Okay. If we had wanted L glucose, what would we do? We'd flip all of these, right? Yeah. So we flip all of these OHs, and that would give us the L. If we wanted L-galactose, what would we do? Yeah, switch them all. Okay? If you just switch the last one, you'll actually get L of a different sugar. Okay? All right. Fantastic. This right here is actually the different monomers that you would see in a chart form. The projector's kind of small and low resolution, so you can't really see it. But it's on the PowerPoints online. It's also in your book. Okay? So you can blow it up as much as you want from those. All right. Notice again, though, that we have here the different lengths of the sugar. So this is glyceraldehyde up here. This is ethros. This right here is ribose. And then this over here is allose. And so we have actually the different links in each of these. Okay. And again, this shows you the D's. By convention, you don't usually list the sugars as both D and L. Why would you not bother listing one? Exactly. You don't need it, right? Yeah, you can just... yeah. So... also, D is the sugar that typically is used in biology, and L's are very rarely used. Okay. All right. So let's talk about some of the important monosaccharides and their importance. We have glucose here. This is the most abundant monosaccharide found in nature. Also called dextrose, or blood sugar. This is typically what you're measuring when you're talking about blood sugars, because this is the form of sugar that your body needs to use. This is the form of sugar that provides your brain with all of its energy. Okay? If you had your blood completely filled with fructose, no glucose at all, for some reason you couldn't convert it to glucose, you would die. Okay? Your brain would actually die. Right? There's actually a lot of theories that that's actually a major part of neurodegenerative diseases, is that you can't get glucose supply to parts of your brain, so they begin dying. Okay? It's going to be found in fruits, vegetables. It's also found in corn syrup. It's found in many, many, many places because glucose is the most common sugar used in nature. Not the most common sugar you can buy, though, by the way, if you're wondering. If you go to the store and grab table sugar, you may not actually get glucose. Okay? Glucose can be broken down inside cells to produce energy through a process. This is going to be called glycolysis. Notice, by the way, gluco, so glyco, right? Lysis, cutting, breaking. So we're breaking down sugars to produce energy, okay? Specifically glucose. Diabetics are going to have difficulty getting glucose from the bloodstream into their cells so that glycolysis can occur. This is actually one of the reasons that they have to be regularly monitored for their blood glucose levels. And then you'd have to supplement perhaps insulin so that they'd start intaking the glucose better. Okay? Specifically, people with diabetes, there's actually quite a few forms, by the way, that all involve not being able to regulate the sugars in your body properly. The most common form is going to be an insulin-resistant form, where their insulin is no longer doing what it should because they're actually built up resistance to it. Okay. The glucose is also going to be a sugar unit in sucrose. Sucrose being table sugar, that is actually a disaccharide, right?
Glucose or fructose? Glucose or fructose?
Yeah, fructose. You got it. Yeah, you got it. Lactose. This is a milk sugar. And this has the glucose in it. Amylose, amylopectin, glycogen, and cellulose. Anybody know what glycogen typically is found? Yeah, liver, muscles. It's the form you usually store this in. Okay, that's why if you ever see anybody seen somebody that drinks a lot of alcohol, and they have like a beer belly, that's like hard, right? You know why it's actually different? It's hard and it's not soft like fat. Is because a lot of that's actually glycogen. Yeah. So that's actually your glycogen stores. They just have too much of it in that case. Yeah. So it's not actually fat. Glycogen. Cellulose. Anybody know what cellulose is?
Plants.
Yes. Plants. You got it. You got it. Wood, plant stalks, that sort of thing. Yeah. Yep. All right. So let's talk a bit about galactose. So galactose can be found combined with glucose and disaccharide lactose. This is going to be present in milk and other dairy products. Most animals are lactose intolerant to some degree. Some humans have built up some degree of lactose tolerance. And that's why we can drink milk, even in adulthood. Galactose has a single chiral center, carbon 4, that's arranged opposite of that of glucose. So it's only one chiral center, carbon 4, that is flipped. The diisomers that differ in one chiral center, these are typically called epimers. So this is an epimer of glucose. So galactose, an epimer of glucose. The body can convert galactose into glucose. This is done with an enzyme that's called epimerase. I know. Very imaginative, right? Literally, epimers. ACE. So epimer, ACE. An enzyme that converts epimers. Okay. Yeah. All right, so this is mannose. Mannose is going to be a monosaccharide, most commonly notably found in cranberries. It's not easily absorbed by the body. Cranberry juice has actually been promoted for the treatment of urinary tract infections, UTIs. It's actually due to the excess mannose being flushed through the urinary tract. And so recent studies have now actually questioned this practice and questioned whether this actually functions due to that, or if this is actually meaningful treatment. But this has been widely promoted and used for a long time. So, fructose. As an American, this is the American sugar, if you will. This is the sugar that's in everything, all over the place, all the time. Anybody know why it's in everything in the United States of America? It's heavily subsidized by the government. So the government pays large amounts for corn and specifically fructose to end up being made. So, yeah. So ketose, fructose, is going to be referred to as fruit sugar or levulose. It's actually found in fruits, vegetables, and honey. And so that's honey right there. It does have fructose in it. In combination with glucose, it actually gives us the disaccharide sucrose. That's your table sugar. That's the stereotypical table sugar. There is actually some issues with just saying all table sugar is X, because technically you can put any sugar in the table shaker. And there are people that actually sell other sugars as table sugars. But this is the stereotypical table sugar. And it has the table sugar flavor that you may be used to. That exact sweetness that you'd be expecting. Even though it's not an epimer of glucose, the fructose can actually be broken down for energy production in the body and converted to glucose, or it can be broken down and give you ATP. Not directly by your brain, though. Ribose, this is the pentose. Pentose, pent meaning 5 carbon. And so ribose and 2-deoxyribose are actually going to be part of the nucleic acids that make up our genetic material. DNA, what's the D stand for? Anybody? Yeah, it actually stands for this, deoxyribose, doesn't it? Yeah, specifically the deoxy portion, RNA stands for?
Ribose.
Yeah, ribose. Ribonucleic acid, right? Okay. Deoxyribonucleic acid. Okay. And so, again, ribonucleic acid, RNA, deoxyribonucleic acid, DNA. Again, it incorporates the sugars, ribose, deoxyribose, into the structure. It's specifically the backbone structure. And so it has phosphate. This is a ring. And then jutting off of there into the center is the amino portion. The nucleic acid, excuse me. The base portion. So structurally, the only difference between the two pentoses is actually the absence of an oxygen right here. And so that is actually the only difference between these two. It's specifically carbon 2 here. Ribose is actually found in the vitamin riboflavin and other biologically important molecules. It is extensively used throughout the body, and that's because ATP, ADP, these all have ribose. Okay? And then the D versions of those, did you guys know there's a D version? No? Okay. So anybody ever heard of ATP? Okay. So ATP would actually be the ATP that only has the monophosphate, so AMP, would actually be the one that would go into RNA. Okay? And then you put a small d in front of it, and so that would be DAMP. That would actually be the one that is the deoxy one and go into DNA. Okay? And so for all the ATP, there's actually GTP. Everybody know the DNA sequence? Anybody ever done the letter matching? GC? AT? Yeah? Everybody's had that in their biology classes back in the day? Probably when you were a kid? Okay. So those are actually the bases. Okay? Those are the bases that pair up in the center, right? And so the backbone of this is actually the sugars with a phosphate. And so that ATP you use for energy, that's actually what you would put in to make RNA. The deoxy version is what you would put in to make DNA. Okay? There's also GTP, CTP, UTP, etc. Okay? And those are all the different bases that you would use for DNA. You can actually use those for energy just like you could ATP. Okay? Glucose and diabetes. Let's talk a bit more about that. So glucose levels in your blood and cells are going to be strictly regulated by the signaling hormones. There's actually a couple that work to signal on glucose. When blood glucose levels rise after a meal, the hormone insulin is going to signal cells to take up glucose where it's broken down or stored. Keep in mind that insulin actually works differently depending on the part of your body that you're looking at. Okay? But in the places where it's going to be broken down and stored, it's going to tell those parts to take up glucose after a meal. Okay? So during periods of fasting or sleep, the hormone glucagon is going to signal the liver. This is going to make glucose and send it into the bloodstream. Specifically, it usually converts glycogen into glucose. And that's where you use your stores of this glucose, in the form of glycogen typically, to go ahead and put blood sugar back into your body. Okay? All right, so anybody have any questions about that so far? Does anybody know what happens if you have low blood sugar? No?
Really tired, really fatigued.
Yes, really tired, really fatigued, especially mentally. Okay? Your body can actually use other things to produce energy, like its fats. Your brain, that's it. It needs the glucose. And so mentally especially, you'll feel fatigued and really tired first. Bodily, though, you'll also start experiencing fatigue and tiredness a little bit. Then you're going to also have issues with if you get more and more depleted, what ends up happening? Anybody? Anybody know what happens to diabetics oftentimes?
Eggs.
If they don't get treatment. They go unconscious into a coma, right? Yeah, and so you'll actually have neural degradation. You can actually have brain death from that, and parts of your brain die. You also have issues with your heart because your heart primarily uses glucose as well to power it. And so you can actually have heart damage as well from your varying levels of glucose. Okay? Okay, so during periods of fasting and sleep, again, you're going to get a signal to boost up your glucose levels, right? And you're going to ideally keep a fairly even level with your usage, right? Oftentimes people have problems with this, so almost 10% of the U.S. population has diabetes. Diabetes is actually characterized by prolonged periods of high blood sugar. That's the most common form of diabetes that most people in the United States have. The insulin signal to cells is then compromised, so cells do not take up glucose, and so you end up with all the glucose just staying in the blood. It doesn't do a lot of good out there, it doesn't get stored properly, and you have extremely elevated levels of this glucose. So in type 1 diabetes, insulin production is going to be low, and you can supplement insulin, and that's how you typically treat it. They literally inject insulin into their bodies, right? Everybody's aware of that? Okay. Okay. And they mostly make that in pens now, so they don't mess up and inject too much insulin. Anybody know what happens if you inject too much insulin? Yeah. Yeah, you'll feel very, very sick. And again, you can go into coma. Because what's going to happen is all your glucose is going to get sucked up into your liver and your glucose-using cells, and then you have no blood glucose, right? Okay. You can actually counteract that by just flooding the person with glucose and keeping them at a level with glucose. Yeah. But it does cause major problems, and that's not really a great way to do that. All right. You can also use the opposite hormone, glucagon, to signal the other pathway as well, have them fight it out. There's also a brand new treatment method that has actually a blood sugar sensitive form of insulin that actually will turn itself on and off on its own, depending on the blood sugar levels in the blood, which is really cool. It is so much better. We'll see when that comes out. So in type 2 diabetes, the insulin signal is not received. Up to 95% of cases of diabetes are actually this type. This is the type that typically is the one that gets trained up over a lifetime of eating the wrong diet and exercise, right? Usually. But it is actually preemptively also a genetic condition. You are more likely to get it depending on your genetics. Okay? Like many diseases. So this is the most common. This can often be controlled with diet, exercise, weight loss, and medication. In fact, there's some really cool treatments that have shown to have some really amazing effects where a doctor very carefully monitors your diet, takes you down to zero carb diet, an absolute zero carb diet near fasting conditions for a couple months. And then they've actually been able to reset something like 30% of cases that they've treated this way, which is pretty amazing, right? Bad enough, if you get bad enough on type 2 diabetes, you can have to take various different medications, all the way up to taking insulin. Okay. Type 1, most cases of type 1, there's not a lot you can do. There's just something wrong with your pathway that makes insulin usually. But anyways, type 2, usually something that can be done. Okay. So the reactions of monosaccharides, your linear structures don't really show how most monosaccharides are actually structured when they're in biological systems. Okay. The vast majority of monosaccharide in your blood, in biological systems, really even in water, are just not going to be linear at all. Okay? They're going to be rings typically. So the pentoses and hexoses, these are again the 5 and 6 carbon monosaccharides, will bend back onto themselves and form a ring that includes that oxygen. Okay? When opposite charges on different functional groups within the monosaccharide attract strongly enough, new bonds are going to be formed and other bonds are going to be broken. And this is actually a factor of you being in water. So the carbonyl group that's going to be present on your aldehydes and ketones right here, see how we have our aldehyde here, is going to react with the alcohol here, because this has a partial positive charge on this carbon here. Can everybody see that okay? Is everybody okay with that part? That this is partially positive? Because that oxygen's going to be pulling electrons away from it, right? Okay. So what ends up happening is if you can make a 5 or 6 membered ring, remember that those are very stable, right? And so this is going to actually wrap around to the end here. This alcohol is actually going to give its electrons to that carbon that's a little positive. And it's going to form this, which is the hemiacetal. So what ends up happening is these electrons get pulled into a bond. And then you're going to get an H plus on here, and this H lost. Okay? So this is a hemiacetal. What does hemi mean, by the way? Okay, hemisphere. What does that mean? Okay, a sphere means a sphere, right? So if we're on half of the globe, hemisphere, that means it is half the sphere, right? Okay, so the full acetyl would actually have another one of these OR groups off of here. And that would be a hemiacetal right here. So hemiacetal is because this doesn't have an R. Okay, and so keep in mind this is the bonds formed. This was the bonds broken right here. Okay. And so we've wrapped back around. We have the oxygen that's in the ring now, and it's going to look like this right here. So this right here is D-glucose. It's going to undergo that reaction, which is going to form our hemiacetal right here. Notice we can actually have two forms of this. One ends up with the oxygen above the ring. One ends up with it below. Okay. Specifically, it's going to be on the same side as this group. That CH2OH. Remember, that's this one here. Okay. That right there is beta. The easy way to kind of remember that is beta. See how the tail lifts it above the line, doesn't it? So it goes higher. Above. Okay. This right here is going to be where your OH here, that is from the aldehyde, is going to be on the opposite side of this group. Okay. All right. And that's going to give you the alpha D-glucose. Again, the D because this is on the right side here, right? Alpha because this OH is below the ring. Beta because it's above the ring. Okay. And again, these are both hemiacetals right now because they don't have another R group attached right here, right? Okay. All right, so the two structures that are possible during ring formation, the oxygen and the hydroxyl group on carbon 5, can form a bond on either the top or bottom of the carbonyl. These two are in inconvertible forms. These are called anomers. When you're in water, any acidic or basic solution, or blood, you are actually going to have continuous interchange between the two. Okay? It's only when you're in conditions where it's reacted that and it fixed it that you're not going to have change between the two. Okay? So in many monosaccharides, the carbonyl carbon that reacts to form the hemiacetal in the reaction is actually referred to as the anomeric carbon because it can form different forms. Anomers specifically, right? Anomeric carbon forms different anomers when it does your ring reaction. Okay? So note, a ring formed, the anomeric carbon is only a carbon bond directly between two oxygen atoms. So it's very easy to find because all you gotta do is just find the one that has two oxygens connected to it. Everybody said okay? 1, 2. So it always ends up with 2 oxygens attached. So the 2 hemiacetyl anomers of D-glucose are going to be alpha and beta. Those are going to be your 2 anomers. The anomers are going to be distinguished by, again, the position of the OH group on the anomeric carbon relative to the position on the carbon outside the group. Okay? If they're on the same side, that's beta. If they're on opposite sides, that's alpha. Opposite sides of the ring, same side of the ring. Okay. All right, so your 6-membered ring form of D-isomers. This is going to be called pyranose or pyranose. Again, this is pyranose. It has 6 carbons. It's always drawn on the top side of the ring. In the alpha anomer, the OH on the anomeric carbon is going to be trans to carbon 6. Opposite sides. On the beta anomer, they're going to be cis. They're going to be on the same side of the ring. Let's go ahead and look at drawing these. So the first thing you want to do is you want to go ahead and create a scaffold of your ring. So you want to draw a 6-membered ring with one corner of it, the furthest to the right, so back and right, being the oxygen. Okay. This right here is going to be that ring closing OH. Okay. And so this up here on the top left here on carbon 5, numbering carbons 1 here on the bottom right that attaches the oxygen, goes around to 5 here, and then this 6-1 goes on the top, and you always orient that upwards. Okay? Okay, everybody good with the ring so far? All right. Okay, so what's going to happen then is you're going to fill in your OHs either as on top or on the bottom, because this is going to dictate alpha or beta, right? Okay, so if you're alpha, then it would be down here for the OH, and it'd be the opposite side of this carbon, right? If you're beta, it's going to be on the top, same side. Everybody good with that so far? Okay. All right, so the next part, you need to fill in these hydroxides, the OHs, based on what type of sugar you have. So for example, here we have galactose. And so we would go through and look at this, and we need to orient these so they're on the correct side from each other in each case. Okay? So for example, if this one here was on the right side, that's typically going to be your, so these are like this. Everybody see that? Okay. Okay. All right. So this right here is your D-galactose. That one's on top there. Is it okay? This one here, D-galactose, this is on bottom. Okay. Again, which one's the aldehyde oxygen? Does anybody remember? Which one was this one? Yeah, it's the number 1, isn't it? Yeah. So it's always this one is your top of your ring. This one right here is always that last OH. Okay? All right. And the important part to get is make sure you orient these the correct sides. Okay? That's always going to be your important part. Keep in mind that this one right here actually gets included in the ring. Okay? And so the rest of them are the important ones to keep track of, because this one's chirality actually disappears. Right? This one actually you don't get a choice. Mm-hmm.
We have to have the Fisher projection so that we can know like with the H and the H.
So you don't necessarily, but you would need to know what the chirality set for that sugar is. So you need to know is
I have to memorize each sugar.
you would, if you were to do it without the Fisher projection, yes, but I'm never going to make you do that. That's just not necessary for this. Yeah. Okay. So if we look here, what we notice is that this should be on opposite sides with this one. And so these are opposite sides. This next one's opposite side right there. See that? So down, up, down. Down, up, down. Okay. So again, the right side is always going to be placed on the bottom of the ring. The left side is placed on the top of the ring. Okay. So let's talk a bit more about reactions. Your D-fructose is going to contain a ketone group instead of an aldehyde. So there's again several hydroxyl groups. The OH on carbon 5 can curl around and react with the carbonyl position on carbon 2. It's going to allow actually 2 possible ring structures. 4 carbons and an oxygen. This will give you a 5-membered ring. This is actually called furanose. And 2 of the carbons, C1 and C6, are going to remain outside of the ring. So the alpha and beta anomers are going to again be determined by the OH group on the anomeric carbon, and this is going to again be related to that carbon 6. So if it's on the same side as the carbon 6, beta, right? So on opposite sides, alpha. Okay, this is what that looks like. So notice that instead of having that carbonyl here, right, like we did for the aldehyde, it's actually down here for the ketone. Okay? So our ring closing happens here, doesn't it? Everybody say okay? Okay. So again, this right here is this one. This right here is this one. Okay. And so that actually ends up giving you the 5-membered ring when we do that reaction. So that gives you the 5-membered ring here. Okay. Okay, and again, the alpha is it being opposite sides here. The beta is the same side. Okay. Anybody have any questions on that? Did I lose anybody there? All right. So your carbonyl group and the aldoses, this can also undergo the organic oxidation reduction reaction. So this can actually occur via adding oxygen or losing hydrogen during the oxidation, or by adding hydrogen or losing oxygen during the reduction. And so right here we see the oxidation, where we've taken the aldehyde, we've reacted it to the carboxylic acid, and so that is an oxidation there. Okay. Right here we see the alcohol being formed from the aldehyde, and that's the reduction right there. This is actually what gives you sugar acids and sugar alcohols. Anybody ever heard of a sugar alcohol before? No? Okay. The sugar alcohols are used a lot for the artificial sweeteners, the things that you can't digest but taste like sugar. Yeah. Okay. So let's talk a bit about Benedict's test. This is actually going to test for the presence of an aldose in solution. The copper 2 plus is going to be soluble. This will color your initial reaction, solution blue. You then put it in with the aldose here. And the aldehyde group is going to undergo oxidation while reducing your copper ions to copper 1 plus. Resulting copper 1 plus oxide is actually not soluble. It's actually going to produce a very sort of brownish red, or brick red is often called, precipitate that will fall out of solution. Okay? Anybody ever done this Benedict's test, by the way? I know they do that a lot in the biology, like 100 level. No? You guys never put a solution in a test for the presence of sugars? Huh? Okay. I'll have to ask what exactly that's done. Okay, so because the aldoses are easily oxidized, they can actually serve as reducing agents, and these are going to be referred to as reducing sugars. And so remember, the aldoses are the ones with the aldehydes on them, right? So this works with the aldehyde functional group. on the aldoses. So fructose and other ketoses are reducing sugars. Even though they don't contain an aldehyde group, this is because they can rearrange to aldoses in the presence of an oxidizing agent. All the monosaccharides are going to be reducing sugars because they can always rearrange to form the aldehyde. Okay? And so this right here, again, is the blue Benedict's agent. We put this in with glucose here, and this is going to reduce the copper here, oxidize this group right here. Notice that gives you the carboxylic acid form, or carboxyl sugar. And that's going to give you the brick red solid. This is kind of what that looks like. We have the blue solution in with our sugar. They're all clear. Cool. We put it together. Notice this is now opaque. You can't see through it. And it's a brownish red color, right? Like a brick red. And that's going to give you the positive test for reducing sugar, which is going to be all monosaccharides or anyone that has an aldehyde. Okay? So the aldoses and ketoses, this can also be reduced to an alcohol when the carbonyl reacts with a hydrogen. Sugar alcohols are going to still have sweet tastes, but they do not have the calories that are found in monosaccharides because your body cannot properly digest them. Okay? Sugar alcohols are going to be things like sorbitol, xylitol, erythritol. These are going to be added to low-calorie drinks and candies, and this is how you usually get things that are sugar-free. This is one of those. Okay, this is not Splenda, by the way. This is one of the options. So an enzyme called the aldose reductase actually acts to reduce the excess glucose to sugar alcohol. This is sorbitol specifically, which at high concentration can actually contribute to cataracts. So if you have this in high concentration, then it can attribute to cataracts, form of inflammation of cataracts. Keep in mind, though, that when I talk about these high concentration things, that everything is a poison at the correct dose. Okay? The poisoner's motto is that the proper dose is a medicine, the improper dose is a toxin. Okay? Anything biologically active can be either. All right, so even when enclosed in a ring, the anomeric carbon is still going to be reactive because it has all those oxygens bonded to it. Disaccharides are going to be formed when monosaccharides are joined together through the condensation reaction occurring at that anomeric carbon. And so that actually is the spot where you get this disaccharide linkages, the oligosaccharide linkages, and the polysaccharide linkages. Okay? Remember how I was making that big deal about acetyl versus hemiacetyl? Guess what? Okay, so let's talk about the condensation and hydrolysis reactions that's forming and breaking what are called glycosidic bonds. So in a monosaccharide in ring formation, your aromatic carbon is going to have the most reactive OH in the molecule, and that's going to be your C1 and your aldoses. When the hydroxyl on the aromatic carbon reacts with a hydroxyl on another monosaccharide, you're going to have a glycoside forming. And that's where the glycosidic linkage or glycosidic bond comes from. So the bond that links the two is referred to as glycosidic bond. The glycosidic bonds will be what join the monosaccharides together to form longer structures, more complex sugars, i.e. in this case we'll be doing a disaccharide. Okay, so this right here is maltose. And so we have the monosaccharide alpha D-glucose and another alpha D-glucose right here. They're going to link together. This oxygen here is going to react with that carbon here, and that's going to form here, which is two glucose linked together. Notice we actually have an alpha position on here, and so this is referred to as an alpha glycosidic bond. Okay? And that gives you this linkage. Okay. So let's name that glycosidic bonds. We have alpha and beta, of course. Those are always going to be named based off the alpha and beta of this OH group right here, the one on carbon 1. Okay? Notice that this right here is going to be your carbon 1, 2, 3, 4 in this case. That links it up right there. That doesn't have an alpha or beta though, because that's not an anomer. Okay? That one's actually depending on the glucose, whether that would be above or below. Okay, so remember that your monosaccharides has many hydroxyl groups that can undergo condensation. It's actually necessary to specify the carbon atoms that are joined by your glycosidic bond, because you could actually form a bond with another one, not 4. Okay, so in this case right here, we're going to have again an alpha, and then it's going to be 1, 4, because we have a linkage happening between the first carbon's hydroxyl and the 4th carbon on the other ring, right? Okay. And so that's what that looks like again. That's maltose, or maltose. And then right here we have the beta 1,4 glycosidic bond. Notice again beta because it's above the ring this time. Okay. And that's solo bias. So let's talk about a few important disaccharides. We have maltose, lactose, sucrose. So these are 3 of the most common disaccharides here. Right here we have our glucose reacting with another D-glucose. That's going to give us our maltose. We lose water, and the glycosidic bond is an alpha 1,4. Okay. Here we have D-galactose with D-glucose. That gives us our lactose, like in milk. And then again we lose water because it's a dehydration reaction. And this is going to be our glycosidic bond again, and it's going to be a beta 1,4. Okay. And then this is D-glucose with D-fructose. This gives us sucrose. We lose water again. And this is actually a glycosidic bond. This one's actually interesting because it happens between an alpha and beta. So alpha from here, beta from here. And it's the 1, 2 carbons. That would be where the hydroxyl groups are. Okay. So right here is maltose, typically referred to as malt sugar. You guys have probably heard of malt sugar before, right? I imagine everybody's tasted maltose, Tennessee. Okay. Anybody heard of malt liquor? Okay, well, guess what? So again, the malted barley contains high levels of maltose, which is why if you use malted barley, you are making malt liquor, right? Okay. And so the glucose and maltose of the malted barley, this is going to be converted to alcohol by yeast. This is a process called fermentation. You can convert any sugar, almost any sugar, into alcohol via yeast. They just would not be malt liquors in that case. Okay? They're malt liquors in this case because you use malted barley actually. The glycosidic bond is going to be your maltose alpha 1,4 because again, the anomeric carbon is free here. The maltose is a reducing sugar. So again, we have that anomeric carbon here that is free exposed here. And so this one right here would be a reducing sugar. If you were to do that same test that we had referred to before with the copper 2+, we would see that color change going from blue to that red precipitate. Alright, so this is lactose. Again, this can be the milk sugar found in mammalian milk. The intolerance to lactose occurs in people without lactase, which is the enzyme, or a less functional lactase, or they don't make it as much. When lactose remains undigested, intestinal bacteria will break it down. This would produce abdominal gas and cramping because they're overworking that pathway. The glycosidic bond is going to be lactose, is going to be the beta 1,4. Again, be the C1 of beta galactose with the C4 of your glucose here. And again, it would be beta, same side as that carbon. The anomeric carbon on the glucose is again free, and it's not the glycosidic bond. It's not included in that. So again, lactose would be another reducing sugar, right?
Can it be carbon one, the glucose, and carbon 4, the galactose?
What do you mean?
Here the linkage is between carbon one.
Oh, no, it has to be one here. Yeah, yeah. If you were to swap those, that would no longer be lactose. Okay. So again, this is sucrose here. This is actually the first not reducing sugar. Finally, right? Finally. So sucrose, again, is going to be your abundant disaccharide in nature. It's why it's typically used in things like table sugar and the most common sugar. It's because it shows up a lot in sugar cane and sugar beets. And so we extract those from sugar cane and sugar beets, though fructose is becoming very common sugar to find in products now, right? Because corn. When glucose and fructose join in an alpha beta 1,2 glycosidic bond, sucrose is going to be formed. And so here we see the alpha 1. Everybody see that okay? And then this one is the beta and 2. Okay. This is the second carbon because this was the ketone, right? See that? The one carbon's right there. Okay. And again, it would not be lactose if it was swapped. And you wouldn't have it too here anyways, but yeah. And so there's no free enantiomer carbon. They're both joined into this new linkage right here, that hemiacetyl, right? I'm sorry, acetyl in this case. And both of them are acetyls in this case. And so this is not reducing sugar because it has no exposed aldehyde or ketone, right? Specifically aldehyde being important. All right, so this is sweetness. In relative terms, different sugars actually have different sweetnesses because they trigger our tongue's sensory differently, okay? The artificial sugars typically have much higher sweetnesses. But for example, fructose is commonly used in sweet products because it has one of the highest natural sugar sweetnesses. Okay? And so it's very, very potent sweetness, right? And gives a very, very potent sweet flavor. Right here, this is the inverted sugar, the hydrolyzed sucrose. This is typically found in honey. So right here, sucrose. You can see it's a 100. This is why it's commonly used for table sugar because it's very sweet. But it's not as sweet as fructose. Okay. This right here is then glucose. This is why you typically don't find that a lot in things that are as sweet. Like fruits are kind of moderately sweet, right? Not as sweet as like a soda, right? Yeah. Yeah. Okay. And then milk, barely sweet at all, right? Yeah. And that's all the relative sugar tastings. This right here is actually some other sweeteners to give you an idea. This is actually Splenda here. That's sucralose. That's actually a chlorinated disaccharide. So they've added chlorines across that disaccharide so it can no longer be digested. And that has a sweetness index of 60,000. So 600 times table sugar, right? And so if you use the same amount of sugar, table sugar, and Splenda in a recipe, guess what happens? It's horrible, right? Anybody ever done that? Or had somebody do that? Yeah. Okay. Anyways, this is equal here. Truvia, sweet and lows right there. That's that pink one. Anybody have any questions on those? Notice overall they tend to be way, 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 way sweeter. Okay, that's why they give that cloying, sickly sweet if you use those packets. Okay, let's talk about polysaccharides. So glucose, again, can be stored in both plants and animals. It's going to use starch and glycogen, respectively. It's going to be connecting the alpha glucose units through glycosidic bonds. And so we'll have those repeated glycosidic bonds in long, 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 long chains. Okay? The connections between the beta glucose units is going to produce molecules. For example, cellulose is going to be where we have repeated beta glucose units. This is the structural material in plants. Polysaccharides are going to be very large molecules. They're going to consist of monosaccharides bound through their anomeric carbon atoms repeatedly over and over. And so you can have polysaccharides that are tens of thousands long. Okay. Polysaccharides do not contain a sufficient number of reducing ends to give a positive Benedict's test. So if you were to take a fairly fresh polysaccharide that you have not introduced your saliva to, and you were to put it in with a Benedict's test, it would stay mostly blue, give you very little of the red. Okay? Because it would mostly not be reducing in. Most of the reducing ends are actually consumed and used and hidden. Very slowly, it would work on it and break it down. Okay? However, if you get starch on your hands or wheat on your hands or any of these things on your hands, sugars on your hands, and then lick your fingers, what happens? Anybody know where you start digesting sugars and starch? Mouth. Yeah, saliva. And so that's actually why if you don't clean your hands at all and don't do anything with them, they aren't sticky after they get covered in sugar. But if you lick your fingers, they get sticky. Now, the disturbing part is you realize you have saliva on your fingers most of the time. So anytime you get something sugar on your fingers, if it gets sticky, know that you probably had saliva on your fingers, right? That's why it got sticky. You touch the wheel on your car and get sticky, you had it covered in saliva. You just didn't realize. Okay. So let's talk a bit about some more storage polysaccharides. We have amylose and amylopectin. So starch is going to be the glucose storage polysaccharide that will accumulate in small granules in plant cells. This is the one you're probably thinking of when you think potato, right? Okay. And you know what this looks like because you've bought starch and you've seen starch. Okay. Very small granules, straight chain for the most part. So starch is actually a mixture of two different forms. We have amylose and amylopectin. So amylose is going to be what makes up 20% of the starch. It's going to be made up of 250 to 4,000 D-glucose units. These D-glucose units will be bound with an alpha-1,4 bond, glycosidic bond. And this is going to give you a continuous long chain. Okay? So this gives you your straight long chain that would go off into infinity. Okay? You can make it basically as long as you want. The long chains of amylose tend to coil up a bit like a spring. Okay? Kind of like DNA does. DNA also coils, right? Or your hose at your house also coils, etc. Amylopectin, this is going to make up about 80% of plant starch. It's also going to contain D-glucose units. They're going to be connected this time by an alpha-1,4 glycosidic bonds. But every 25 glucose units, you're going along that linear chain that we talked about before, you're going to get a second chain that will branch off. So you got your long chain, and then you have branching chains coming off it every about 25 or so glucose units. Okay? And so these will actually be alpha-1,6 glycosidic bonds. So that's how you get the branching, is it actually attaches to the 6 instead of the 4, right? So it gives you the branch off. And that's every 25. Okay? When we eat starch, our digestive system actually breaks it down to glucose units for use in our body. And so we basically just chop away at the starch, freeing the glucose. Okay? This right here is actually a straight chain starch right there. And see how we see the coiling right there. Everybody good with that? Okay. We'll call that a day, I guess. And this right here, again, is the branchment. And there's the branching. All right. You all have a wonderful day. We'll start on glycogen next time. That's the one we carry around. Ooh. It's good stuff.
I'm just